My name is Stephen Hunt. I'm a physician scientist at the University of Pennsylvania on faculty here, and also one of the founders and co-directors of the Penn Image-Guided Interventions Lab. What my job is, what an interventional oncologist does is, think about the world of cancer care. You have medical oncologists who dole out poison, chemotherapy, right? They're going to poison the cancer. And they're kind of the queen on the chessboard, right? You have the surgical oncologist who are going to cut it out. They're going to literally take a knife and cut the thing out, right? And I call them the king on the chessboard. They're powerful, but man, the king wears red. I mean, it's a bloody mess, right? You have the radiation oncologist. I think of them as, they're going to deliver radiation to the tumor, right? I think of them as the priests. They wave their hands, and through some kind of magic that none of us can see, you know, the cancer goes away, right? But like all religion, it has terrible side effects. So they get radiation pneumonitis, they get all these complications of the radiation. So then what you have is, so that's medical oncologist, surgical oncologist, radiation oncologist, and then there's the fourth, and we're the most recent branch of oncology, and we're called interventional oncologists. So what we do is these local regional therapies. I put a catheter, instead of delivering chemotherapy to the entire body and poisoning all the normal structures, I put a catheter right up into the vessel feeding the tumor and deliver chemotherapy and beads to block the vasculature in the tumor, in the liver, for example. That's called chemoembolization. Or I put a little catheter up, so that's very akin to what the medical oncologist is doing, except for instead of delivering chemo to the whole body, I'm delivering chemo just to the tumor. Or I do what the radiation oncologists do. I do radiation. But again, I put a catheter up just into the vessels feeding the tumor, and I deliver these little glass spheres that come all the way from Australia. Okay, they make them in a nuclear reactor over there. They ship them over at a cost of about $50,000 for a treatment. They ship them, I have to order it weeks ahead of time. They ship it over. I put the catheter up, take this one-time dose, this poor patient's insurance is paying $50,000 for it, and put that little dose into the tumor through a catheter. So again, instead of being radiation to the whole body, radiation just to the tumor. So you see how it's precision medicine. It's very targeted. Or in terms of like the surgeon. So the surgeon cuts it out. Well, I put a needle into it and I freeze it, cryoablation. So a needle goes in there, a little ice ball forms around the edge of the needle by flowing a very cold gas like argon gas or helium gas through the needle. That freezes, makes this big ice ball and kills a tumor by freezing. Or I put a needle into it and microwave. It generates a microwave, the microwave cooks the tumor just like it would in your microwave at home. So those are called ablation technologies. So I have embolization technologies, radioembolization, chemoembolization, ablation. The research I do a lot of is also on focused ultrasound. So you can also use energy from ultrasound. But we've developed a device in our lab called electrochemical ablation, where we put electrodes in and then actually a very low current. Not enough to cook the tumor, but enough to cause a chemical imbalance in the tumor, causes the cancer cells to die from that. So we'll have some papers coming out this month on that. Always think of new ways of killing cancer, and that's what my lab is focused on. There's about, at this point, about 25 people in our lab treating cancer, and that's the new field of interventional oncology. We started the PIGI Lab in 2012. Yeah, that's when we first started it. So we're now 5 years in. So the three guys started it, me, Terrence Gade, and Greg Nadolski. We were all residents together in radiology residency here. And basically our first year, we said, there's no one doing real basic science research. We want to do it. We came up with a logo. We were virtual. All we were was a logo. We had no money, no space, no real reality other than we're a lab, right? First thing we did was go write for grants. We got some grants. We were housed in other people's labs doing experiments, but with money that we had brought in. So they let us do the experiments in their lab. You know, in all of those things, I think science requires a big community of finding our way in the dark. There's a lot of serendipity to things as well. You think you're headed in one direction and you find something totally different. If you have the presence of mind and the observational capacity to say, I'm not going to throw away that piece of data. I'm just going to try to really figure out what it means. It wasn't what I expected, but what does it mean? And so you turn your failures into successes that way. The PIGI Lab stands for, it's a little bit of a nod to my past, obviously, PIGI, for the fact that I grew up on a pig farm, but it stands for Penn Image-Guided Interventions Lab. And so image-guided interventions is what we do as interventional radiologists. And really, what we're about is trying to harness, innovation and bring it to the clinic as quickly as possible. So everything that we do in the lab, in animals and in cells, we then try to have a clinical trial that it develops into for our patients. So for example, someone came into me today, relatively young person with new diagnosis of liver cancer. And they want to be as aggressive as possible. They want to try any kind of experimental therapies that might be available. So one of the things that when we were studying in our lab, this process that we do called embolization, where we put the little beads in and stop the blood supply to the tumor, we found that some cells survive. They survive this environment through a process called autophagy. Autophagy. And what autophagy is that if you cut off all the blood supply to a tumor, and most of the tumor will die, some cells start digesting their internal organelles, like their lysosome and the endosome and all those kind of things. And they stay alive long enough by kind of eating the trash that when the blood supply comes back, they're able to regrow. And so there's a drug called chloroquine and hydroxychloroquine that is used against malaria, but it also turns to inhibit autophagy. So we showed first that it would do it with cells, liver cancer cells. We're growing the patient's liver cancer cells. Then we showed that in this rat model of liver cancer, it killed, indeed, when you combined it with embolization, that it made the regrowth rates, the rates in which the cancer regrows, to be less. So now we have clinical trials. So this patient today, I signed him up for the clinical trial. That's something that came directly from, we started this research in the lab, we tried it out in cells, moved it into animals, now we're in a phase 1 trial in people. Phase 1-2 trial in people. So that's the kind of translation, and that's what the notion of people always ask, what's the point of being an MD PhD? Why don't you just choose what you want to do with your life? Be an MD, be a PhD. But see, a PhD wouldn't understand the clinical context and like what are real, I mean, they could to some degree, but they don't have that visceral, it's not their patients, right? And for me, and as an MD, I wouldn't understand the science enough if I wasn't really in the lab. And there are MDs who devote their life to research, so I'm not saying that, but I'm saying, you know, what the MD PhD allows you to do is, on the one hand, you can talk to science. On the other hand, you have that clinical knowledge as well, and sometimes a clinical practice like me, where when I think about going tonight, when I'm going to have to leave my family in my nice house, instead of watching cartoons with the kids or whatever Friday night, I'm going to have to go in and embolize these rats. What I'm thinking about is that guy from earlier today. I'm thinking about that patient from earlier today who's, if this experiments work, and he's trying it out, he's choosing to be one of these rats. He's choosing to try out this novel therapy that maybe it'll work, maybe it won't. I love the joy of discovery. I love asking the questions, coming up with a really unique question where you're like, when we know the answer to that, it's going to change the way people think about it. That's a lot of fun.
Is that the uniqueness? Or is that
I think that there is a unique, translational labs are few and far between. Which is probably why we got, Terrence got that big grant, and I get to, most people are either focused on the science alone, completely divorced from the clinical space that it's going to go into, or they're focused on, let's say, disease. I want to cure this disease, I'm going to be focused on that, and because of that, they kind of have their blinders set like that. And both of them are valid, by the way, because a lot of science is serendipity. It's not necessarily that I'm going to set off in this direction and I'm going to ask this series of questions and the answers are going to be predetermined and that's going to determine what happens with care for our patients. It's not like that, But in any case, I think that our lab is somewhat unique in that it has this very strong focus on translation. It's basically translate, we have a motto in our lab and it's, translating discoveries today to the clinic tomorrow. It's like basically, let's move fast. And again, move fast, break things. Some things you're going to get them into clinic, they're not going to work out. But guess what? Investing 30 years into them, they still wouldn't have worked out. That's the honest truth, right? And so get it in there as your alpha prototype. Is hydroxychloroquine the best drug for inhibiting autophagy? Maybe not, but here's what hydroxychloroquine is, 4 cents a pill. The dose we need is about 1/3 of what you take for malaria prophylaxis. The stuff's been on the market since 1920 for malaria. So here's a drug that I can tell my patients, hey, it's going to cost you 4 cents a day to take this pill, okay? And it may work. In the lab, it works pretty well, and it may work. The clinical work drives the research side as well because it puts faces to names. So if you look up there is a patient flyer. is actually from a funeral, Fatima Karoma. She literally looked in my eyes as she's dying and said, take care of my children, right? I mean, that's the kind of interaction you have with people that's very powerful, that lasts with you forever, and you never forget them. I think that a big challenge for us has been some of the technologies we deal with and some of the advances we've made are at the intersection. Let's say you have a device, like we've shown that adding a device plus adding an immunotherapy works better than you would ever expect because you generate an immune response with the ablation and then the immunotherapy releases the brakes. So it's like stepping on the gas while releasing the brakes, right? Well, the device companies want to have their piece of information from that trial. The drug companies want to control the trial from their point of view, right? And nobody wants to, everybody wants to be the one who's running the thing and in control. And sometimes it just leads down to breakdown where you're like, you have a perfectly good trial, you're trying to sell it to them, but how do you break down those barriers? And that's, again, I get back to, Sean's trying to encourage breaking down of those barriers, but that still happens at the commercial level and even within the institution. They say, the oncologists say, we don't want you giving out that drug. Even though you're an interventional oncologist, that's something medical oncologists should be doing, not you, right? But it's already been shown preclinically that if you take those same drugs and inject them directly into the tumor in a mouse, you could use 1 microgram of the drug versus 100 micrograms you would need to inject into a mouse to get even close to the same level of response. So by directly injecting into the tumor, like what I would do, it's much more effective. But then the medical oncologists are giving up their whole way of making money, which is to infuse it in the vein, right? So there's those kind of challenges that have to do with, and so I think really what it comes down to is, again, I always ask, how am I going to make this patient's life better? How am I going to improve the situation? But it's also true of your colleagues. How am I going to make the medical oncologist work better with me? And that's a matter of fulfilling the needs that they have of me as well. So sometimes it's just a matter of a simple chest tube. My patient has a big effusion. I've been trying to get them into IR to get a chest tube. You guys are always so booked up, I can't get the patient in. Okay, I'm not on service, but guess what? I'll throw on a pair of scrubs. I'll put the chest tube in. We'll get it done, right? Or I'll call my colleagues and have my fellow do it, So a lot of it is learning how to be collaborative that way. People are afraid of, if you start doing it, then I'm not the novelty anymore. I'm not the one who's, getting that. And the truth is it may be that your publications suffer. The science will be advanced. Right? If everybody's, and the competition will make you better. But people don't want competition. A lot of people avoid competition. And a lot of that has to do with back to that fear of failure. Well, if I let you do this, they look at it as a zero-sum game. And we can look at it in politics too. A lot of people look at it. If you tax this guy, or you have to tax this guy to make this guy's life better, or if you do tax this guy, that's it, he's going to be out, whatever, and all these arguments are spurious, right? What you've seen under Democratic administrations is they often tax more, and yet the market does better. So I don't think that there's a lot of these correlations simply don't exist in the real world. They exist in people's fantasies or in their mind or in their political arguments, But I think that the same thing happens in science. People say, I don't want to collaborate because if I give this piece of information away, it's no longer mine, and now this other person will get credit for it. But of course, the best scientists, that's not how they look at it. Einstein was constantly like, what can you do to if you take my stuff and run with it, then my stuff looks better, right? Then my earliest thing I said about it looks better. But people don't look at it that way. They look at it as like too much like they need to control this and have control over this process. And they want to take the credit. How do you incentivize team science? Well, one of the ways is things like the Cancer X Prize. So if you have a team and you're like, I don't care that I know that the best guy for NMR spectroscopy is at Cornell. It's not here at Penn. I'm not saying that. I'm just saying, for example. So I want that guy on my team. And I think the best geneticist for C. elegans genetics is Sidney Brenner off in the UK. And I think that the best guy for, anyways, you start building your team. And then your team is going, of course, for this $10 million X Prize, right? Now you're breaking down barriers. And so I think that that is an example of a way in which you really can motivate people in a way with the right set of incentives to be doing more team science and to be breaking down those barriers. And that is what the funding mechanism that Sean used for the Parker Institute was, again, about, yeah, you can have this $50 million, this $250 million, but what that means is that you promise to be data sharing, and if you're not, we can pull your grant. So CRISPR is a technique for gene editing. What if you did this in cancer cells? What if you took the patient's cancer cells with a biopsy? We grow some, so what we do is we biopsy a patient's cancer. We take some of that biopsy and we grow it up into mice so that we have a constant colony of mice carrying their cancer around on their backs, okay? And those are going to be the mice that we have to come back and cure if we can ever find the right thing, right? We grow some of the cells in the lab, and we do a bunch of other experiments to the rest of the cells on characterizing the immune response in those cells, characterizing the metabolomics of them. But in any case, we take, and what we do is we made a library of primers, what are called DNA primers, for knocking out 223 genes that correspond with all approved drugs. So all approved FDA drugs funnel through 223 different genetic pathways. So we have made these primer pairs against those 223 pathways, and if you grow up the person's cancer and you knock all of those genes out one by one in the cells, right? And then you say which clones did not grow, which genes were missing. When they're missing, this cell cannot live. So it's almost backwards of what you would normally do is you would sequence and look for the mutations. But that doesn't give you, the problem is it's like a supercomputer. Knowing that a few pieces are missing or don't work, it doesn't tell you what's the one critical piece that take that away and you've destroyed the computer. From the Stanford experience, I started a company where I'd go up and down Sand Hill Road raising money for startups. And most of them were biologics. So there were small molecules that people were using for a given disease state. The two things I heard a lot. One thing was I'd always thought that you raised money based on the chance of success and the size of the market. And so I walked in, I remember one of the first times I was at Three Leaf Ventures. And that's a lot of physicians are at Three Leaf Ventures who are hearing your proposal and commenting on it. And they said, what do you think your chance of success is? And I said, well, I can't exactly ask, what am I going to say? And they said, I'll tell you what your chance of success is. It's 10%. And I said, I don't care if you're Bill Gates, Muhammad Gandhi, the Pope, or my child. If you made it past my secretary, the chance of success is 10%. So you're not here because your chance of success is any higher or any lower than anybody else. They said, all I really care about is another metric. And they threw up a graph and they said, you know what this is? And I said, I don't know, some kind of timeline. I don't really know. They said, TTF, time to failure. That's what I care about. If I'm going to give you my money, fail me tomorrow. That way I can turn it around. But don't drag something out a year, 2 years. And so they said, this is the problem with biologics. I can invest my time 5 years, 10 years. And the whole FDA process and everything else is such that it could drag on forever. In the meantime, my investors have left and gone somewhere else. So we need something that has a much faster turnaround time. So look at devices. And so the big thing was Thursday nights. Thursday nights, Trader Joe's would turn over their flour stock. They would get a new shipment from Peru or wherever their flowers come from, and they would throw away all their flowers. And so we would go get all the flowers, take the beer bottles, which we had washed out on campus, a bunch of beer bottles just from our co-op, we drank a lot, fill them up with water and flowers, and then put flower bombs all around the house. And I remember one time we actually put a flower bomb on Steve Jobs' porch because all of those, people live just in a regular house in Palo Alto, But someone told me, as we were leaving, hey, that was Steve Jobs' house. And I was like, oh, I didn't even know it was any different than any other house, But yeah, we called it flower bombing. So that people would come out in the morning, there would be this bouquet of flowers, in a beer bottle, but hey, beggars can't be choosy, So probably the biggest hurdle to the vast majority of people being entrepreneurs is that they can't commit to taking a risk. And the reason why you see immigrants so often at the forefront of entrepreneurship, in my mind, is because they've already gambled it all. They've already been willing to take that risk of, I could end up with nothing. And in fact, they might have been immunized to that by virtue of the fact that they have started with nothing. And they know that it's okay. It's okay to fail. It's okay to have nothing, because you'll rebuild it. You'll come back again. We do this in our lab all the time. We say, that's a high-risk project. That's fine. I don't mind that it's a high-risk project. What's its time to failure? Is its time to failure 3 years, and we're going to tie up a huge amount of lab resources? Then no, we can't afford to do that. Is its time to failure 3 months, and you're going to go for broke and spend a lot of money, but it's going to be done in 3 months? Do it. Give it a shot. It really just comes down to asking that simple question of, how can I make this person's day better? But you could do that with the lady at the coffee counter,
Right.
right? You smile, you give a $1 tip instead of 30 cents. What did it cost you? It cost you 70 cents.
Right.
Okay, 70 fucking cents. But when you handed them a $1 bill instead of dropping 30 cents in the thing, you made their day better. And it could be that they then go on and make somebody else's day better. It's a pay it forward thing, right? And I'm not, I think that if everybody went around asking how can I make somebody's day better, the world would be a much better place. Yeah, I mean, I think that get involved in, if you have a family member who has a medical condition, I don't care if that medical condition is multiple sclerosis or breast cancer or whatever, get involved with an organization on that and help spread awareness around it. Help them fundraise. If you have labs, obviously, like we have a research foundation, the Penn Image Guided Research Foundation, and people can donate to those kind of 501c3 charities to fund research directly. And I think that you are going to see more of that direct philanthropy because experiments.com and various websites that have been set up to take advantage of crowdfunding of even research. And like we do little things, you can get a cup with our little logo on it, or you can do this or that. But I mean, really what it's about is, most of the gifts we get are from patients who have their lives been affected by one of us treating them. And they say, hey, what can I do to help? Say, oh, give money to the lab. The lab is doing research in it. So, that's a possibility. But I think for the most part, the American Cancer Society, the American Heart Association, all these things, if you have a family member who has that, or friends, or just something that really strikes you as this is a cause I really care about, then obviously give your time, give your money, and help, again, what can I do to make this person's day a little better?